In this lesson, we'll continue our discussion of the cellular level of organization by taking a look at the cytoplasm and then the structure and functions of the non-membranous organelles. So we can see here from this diagram, we have a cell, right, and we looked at the plasma membrane already. We did that in a previous lesson. Everything inside the plasma membrane is known as the cytoplasm, and we divide the cytoplasm into two parts. The watery part is going to be called the cytosol. Sometimes we refer to that as the intracellular fluid. Then suspended in the cytosol are these small little baby organs known as organelles. So we divide the organelles into two parts. The non-membranous organelles, that's going to be the topic of our discussion today, and the membranous organelles we'll do in a subsequent lesson. So we'll see there's the cytoskeleton, all the different fibers of the cytoskeleton, these small finger-like projections known as microvilli. We'll take a look at centrosomes and centrioles, cilia, flagella, and then we'll finish up this lesson looking at the ribosomes. So let's take a look at the cytoplasm. So as I said before, everything inside the cell membrane is going to be known as the cytoplasm, but that excludes the nucleus. We're also going to do that in a separate lesson. And we can divide the cytoplasm, if you want to highlight, into two parts: the cytosol, and then going down a bit, the organelles, right? So the cytosol is also known as the intracellular fluid. We sometimes just call that the ICF. And this has a bunch of things dissolved in it, things like nutrients, maybe like amino acids, fatty acids, glucose, that type of thing, ions, proteins, and even waste products that need to be eliminated from the cell. Typically, the cytosol is high in proteins. This becomes important later in the semester when we look at the function of nerve cells. These proteins are important to us. And there's also the main intracellular ion is potassium. There's always going to be high amounts of potassium, right? Relatively low carbohydrate levels, lipids, amino acids, and sodium. Sodium we want to also keep out. So the organelles that we're going to look at are basically just structures with specific functions. I kind of think of them as little baby organs. So again, we're going to focus on in this lesson on non-membranous organelles. So what does that mean? What am I saying when I say non-membranous? Basically, the organelle itself does not have its own cell membrane. The organelle itself is in direct contact with the watery part known as the cytosol. And we're going to look at, again, cytoskeleton, centrioles, ribosomes, proteosomes, microvilli, cilia, and flagella, right? Not necessarily in that order. In a subsequent lesson, we'll take a look at the membranous organelles. These are organelles that actually have their own plasma membrane, right? So they actually have their own membrane. And we're going to see that includes the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, and the mitochondria. Now, something that we are not going to talk about in this lesson or in this class even, many cells may store substances within the cytoplasm. We call these inclusions, right? These are masses of insoluble materials in the cells. An example would be glycogen. If you remember, glycogen was our stored form of glucose that is stored in the liver and the skeletal muscle. Or even in fat cells would be lipid droplets, right? Fat droplets. So let's begin our discussion of the non-membranous organelles. So the first one is known as the microvilli. Very simple. Whenever you hear villi, think finger-shaped or finger-like projections. They're kind of these projections that look like your fingers, right? So these are small finger-shaped projections of the plasma membrane. Now, by having these finger-like projections, that greatly increases the surface area. That means there's more cell membrane available, and that gives us the benefit, or these cells that have microvilli, the benefit of absorption. Okay, so we typically will see cells with microvilli in the digestive system, because if you think about it, that's where most of the nutrients are being absorbed into the bloodstream. Now, what keeps the microvilli upright are a type of filament. We're going to look at this in more detail in a couple moments, known as microfilaments. These extend up into the core of the microvilli, and they keep the microvilli upright. So take a look here. Here's a picture of our cell. Right? We have a part of the cell. This finger-like projection going up here, right? And then here's another one which is cut and we're looking into it. Here's a third one. Here's a fourth one. These are the microvilli. The two front ones here, they're cut on a frontal section so we can look into it. And if you see these fibers going up, these are the microfilaments, and their job is to keep that upright. If these were to be like collapsed down or folded over, they would be useless to us, right? They wouldn't function. And you can see here that these microvilli gives us much more cell membrane than we would have if this was just a flat top to this cell. Okay, so let's just go back, make sure we got everything. Okay, so micro means small, villi is finger-like projections. They increase the surface area for absorption. That's the function of the microvilli. And as a result, one of the places we see them mostly is going to be in the digestive system. All right, here they are. Here's an actual micrograph showing the microvilli. Look at this. We're magnifying this 30,000 times to see this. So all these fibers going up, they're projecting up into the microvilli, is going to be the microfilaments. It's a great picture. Right, here's an actual micrograph showing the microvilli. Sometimes if you take a look at this, it kind of looks like the side of your toothbrush. If you held your toothbrush up and you kind of looked at your toothbrush from the side, kind of resembles the bristles of a toothbrush. So we sometimes call the microvilli the brush border. It's a term that a lot of anatomists will use, brush border. Okay, so that's the microvilli. The next structure we want to take a look at is going to be the centrosome, and then within the centrosome are going to be centrioles. There'll actually be two centrioles. So let's take a look at the centrosome first. This is a structure that's located next to the nucleus. Just think of the nucleus for now as like the brain of the cell. We're going to talk about that in a subsequent lesson. So the centrosome is located right next to the nucleus, and this is also going to give rise to part of the cytoskeleton known as microtubules, right? We actually see that the microtubules will radiate out off of the centrosome. Okay, and we'll talk about the microtubules in a couple moments because they're part of the cytoskeleton. Let's take a look. So if you take a look over here in the center, this is the nucleus. Again, think of this as the brain of the cell. Right next to it, this kind of football shaped structure right here is the centrosome, and then you'll see two tubes in it. Those are going to be the centrioles. Okay, so the centrosome, again, you'll usually find it right next to the nucleus. Now, what are the centrioles? These are two, right? So they're paired cylindrical tubes located in the centrosome. They are like straws. They're hollow in the middle. Now, when you look at them, you're going to see that they lie perpendicular to one another, right? They're going to be at a 90-degree angle. So let's take a look at that. So you can see over here, if you kind of look here, you'll see this one tube and this tube are 90 degrees to one another. Right, here's another picture. So you see here's one tube, here's another tube here, and you can see it's hollow in the middle. It's hollow in the middle. So before I go back, notice these centrioles, right? This one big straw-like structure here, all of this, and this one here are the centrioles. What are they made of? They are made up of 1, 2, 3 microtubules. So this is like a triplet of microtubules. Then there's another triplet, another triplet, another triplet, here, here, here. And if you count these all up, guys, you'll see there are 9 triplets of microtubules that form this straw. You have this one too, right? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplets of microtubules. So these microtubules are making up the centrioles. We call this a 9 plus 0 array, right? This means that there's 9 triplets of microtubules with nothing in the middle. This 0 means nothing in the middle, and that'll become important in a little while. Okay, so the two centrioles are going to be two like straws, and the straws are made up of microtubules, but there's 9 triplets. So what's the function? The function of these centrioles are important to us during cell division. We'll talk about in subsequent lessons something known as cell division, and part of cell division is mitosis, where the nucleus divides. So these centrioles will help separate the chromosomes. In addition, the centrioles form the bases, right, which is the bottom of the cilia, as well as flagella, which I'm going to talk about in a couple moments. Okay, so again, centrosome is the football-shaped area. Living in the centrosome are two centrioles. They lay 90 degrees to one another, right? So this one here and this one, they're perpendicular. Okay, they're perpendicular, and each centriole is made up of microtubules arranged in triplets, right? 1, 2, 3. This is one triplet, right? And then we have 9 triplets making up one centriole. 1, 2, 3, 4, 5, 6, 7, 8, 9. Right? And then if we did the count of the same thing here, we would have 9. Right? Here's a real picture, so you can see this darker area right here, centrosome. Here's one centriole lying perpendicular to another centriole. I love this picture. You can look right in the tube and you can see almost the triplets over here. Okay, so we got the microvilli, we have the centrosome with the centrioles. The next structures we want to take a look at is the cytoskeleton. So think about your skeletal system, right? My skeletal system. Our bones, we're going to see, helps to maintain the shape and our body strength. Without our bones, we would be like a jellyfish. So think of the cytoskeleton fibers kind of like our bones within our body, right? So these fibers we're going to see, guys, are going to serve as the cell's skeletal system. Okay, it's going to help to provide for cellular shape and strength, and we're going to examine 3 types of fibers: microfilaments, intermediate fibers or intermediate filaments, and then the microtubules. Watch this. These were the microtubules. So this is the part of the cytoskeleton even made up our centrioles. And watch this here. See these fibers going up here? These are the microfilaments. They're part of the cytoskeleton also. So we've actually looked at a couple fibers already. Okay, so let's start with the microfilaments. These are the thinnest of the filaments, and they're made up of a protein. I do want you to know this protein. We're going to use this one a lot this semester, known as actin. Okay, the microfilaments also form something known as the terminal web. This is important to giving structure to the cell. I'll show you a picture of this in a moment. Okay, so the microfilaments are the smallest filaments, right? They're the thinnest filaments. They're made up of a protein known as actin, and they form the structure known as the terminal web. So generally, the microfilaments are going to anchor the cytoskeleton to the integral proteins of the cell membrane. If you remember from when we did the cell membrane, the integral proteins were the proteins inside the cell membrane. Okay, so these actually attach the cytoskeleton to these proteins. They also provide mechanical strength. They also adjust the consistency of the cytosol. So the cytosol is not like a thin, watery type of water. It's a more gelatinous, a kind of a thicker type of water. That's partially due to the microfilaments. And then when we get to the physiology of muscle, right, the anatomy and physiology of muscle tissue, we're going to see that actin is going to interact with thick filaments known as myosin for muscle contraction. So this is something we're going to look at later in the semester. Okay, so what do we need to know for now? Microfilaments are the thinnest, right, the smallest filament. The protein that makes it up, actin. I'll show you the terminal web in a minute. All right, so the microfilaments help to attach or anchor the cytoskeleton to the integral proteins of the cell membrane, right? They're attaching to the cell membrane. They provide mechanical strength. Again, think of this as our skeletal system. They make the cytosol a little bit thicker, right? Like not so watery. And they will, when we get to muscle tissue, we'll see that the thin filament, the actin, is going to interact with myosin during muscle contraction. So all of these fibers here, guys, we looked at them before going up into the microvilli. These are going to be the microfilaments. See this whole network of fibers? It kind of goes around the whole perimeter of the cell on the inside. This is known as the terminal web. Okay, and then you can see more microfilaments down over in here. Right, so the microfilaments are just kind of spread throughout the cytosol. All right, so here's your microfilaments and this structure here called the terminal web. We'll look at this a little bit more when we get to tissues in a couple lessons from now. Again, we saw this picture before, but all these are microfilaments. Here's the actual terminal web now. I didn't want to mention it before, but we could actually see the terminal web here, and then extending up from there are the microfilaments holding up the microvilli. That's such a great picture. Okay. The second fiber we want to look at are the intermediate fibers. So these are a little bit bigger than the microfilaments, but smaller than the microtubules, right? So I put here they're mid-size between microfilaments and the microtubules. Now, there's a lot of different proteins that make up the intermediate filaments. All I want you to know is these are very durable, they're strong. And I thought maybe one example you may know in skin and hair and your nails is something called keratin. This is an example of a intermediate fiber. Okay, and it helps to give strength to our skin cells, our hair cells, and our nails. So the function of the intermediate filaments in general is to strengthen the cell, and again it's part of the cytoskeleton, so it's going to maintain the shape. Okay, so, again, kind of like the skeletal system will do. It also, this is an important one, helps to hold the organelles in place. Right? Think about this: when you stand up, we don't want gravity to take all of our organelles and have them fall to the bottom of the cell. Right, we want the organelles to stay in place, all these little baby organs. So these intermediate filaments help to hold the organelles in place. And they also help to stabilize the cell position. We're going to see this when we get to the tissues chapter, when I show you how cells are connected to one another. Right, we have these specialized attachments at the plasma membrane, so the intermediate fibers become important there. Okay, so what we need to know is these are in between size of the microfilaments and the microtubules. These are stronger, right? These are going to give us strength, right? So they provide the strength to the cell, they maintain the shape, hold the position of the organelles, and then also stabilizes cell position by attaching one cell to another cell, which I'll talk about again when we get to the tissues chapter. Right, so here's our intermediate filaments. These are the ones that are more violet here, purple. Right, you can see they're a little bit bigger. Right. The third type of cytoskeleton fiber, this is going to be the largest one, known as the microtubules. Right, so we saw them before. They make up the centrioles. Remember the triplets? Those were microtubules. So these are large. These are also hollow tubes made up of tubulin. So let me just go back for a second just to make sure everybody's with me on that. So if you take a look here, guys, here's a microtubule. So notice this is also hollow in the center. So this is a tube, right? This is another tube. Here's another tube, right? 3 microtubules with tubes. That triplet is the 3 microtubules. Okay, so these are large hollow tubes. They're made of a protein named tubulin. Like I said before, when we did the centrosome gives rise to the microtubules, so they are attached to the centrosome, and they also, watch this, strengthen the cell. Right? So all three of the tubes that we've looked at, or the fibers I should say, have strengthened the cell. These also help to hold the organelles in place. Cells that can change their shape, like a white blood cell, the microtubules are involved with that. Sometimes, this is interesting, we have to move organelles within the cell, so the microtubules can help to move organelles around. This is a little bit advanced. There's two other proteins called motor proteins that help the microtubules, kinesin and dynein. These are just, they would assist the microtubules with moving the organelles if organelles need to be moved, right? So they kind of do both. They can anchor organelles, but if they need to move organelles, they can move the organelles. The microtubules are going to be involved with cell division, right? They're going to form something known as the spindle apparatus. That's going to help to separate the chromosomes during cell division, and these are going to form the centrioles, which we just saw a little while ago, and the cilia, which what we're going to look at in a little bit. All right, so these microtubules are important. Again, the largest but hollow tubes made of tubulin, right? They attach to the centrosome, they strengthen the cell, they anchor organelles if organelles need to be stationary, but they can move organelles if organelles have to move. In cells that change their shape, they will help to change their shape. Right? They form the centrioles, remember the 9 triplets, and we're going to see in a moment they form another organelle known as the cilia. And here's an example of a microtubule. So this yellowish tube right here, you can see is a hollow tube right there. And it's stabilizing this organelle right here that we'll talk about in a little bit. Here's another microtubule stabilizing this organelle right over here, kind of holding them in a position. Here's another microtubule. We'll see in a little bit later, this is a mitochondria. Okay. The next non-membranous organelle is known as the cilia. Right? So these are slender extensions of the plasma membrane. This is more like hair growing out of your scalp. They're not actually folds of your scalp like the microvilli would be. These are more like just hair projecting out from the plasma membrane. Now their job is going to be to move substances across the cell membrane, right? So they're going to move fluids across the cell membrane. That type of cilia is known, if you want to highlight down here first, as a motile cilia. Right, so these are found in cells, especially in the respiratory system and the reproductive system. Think about this for a second. Let's say you breathed in some dust and the dust got down into your trachea. So thank goodness the cells of your trachea have cilia. They catch that dust, then they beat, right? They move in such a way that they bring the debris back up so that we can cough it out. These are known as motile cilia, right? These are the ones that are going to move substances. In the reproductive tract, if you want to put down as an example, would be the fallopian tube, right, the uterine tube. These help to move the oocyte, the egg, right, the oocyte down towards the uterus. Or if there was conception, the zygote would be the new cell. All right, so these are going to move the oocyte or the zygote down to the uterus. Okay, so these are called motile cilia. Another type of cilia is known as a primary cilia. These don't move things. These more are sensory. They're kind of like feelers, so they're just kind of sticking out and sensing things in the environment. So typically when we think of cilia for our course, we're basically thinking about the motile one. Let's take a look. So you'll see here, guys, here's the cilia projecting up, okay? Kind of like hair growing out. It's not like the microvilli, but notice what's making up the cilia are microtubules. And if you notice down at the base, it's 9 triplets, right? Watch this. 1, 2, 3, 4, 5, 6, 7, 8, 9 triplets. So this is the 9 plus 0 array. But if you notice in the middle, we have the 9 triplets, but then there's 2 microtubules in the middle. We sometimes call that the 9 plus 2. Okay, so you may see that terminology, but this is what the cilia is look like on cross section, right? So we cut into the cilia and we look into it. That's what it looks like. But at the base, down at the bottom, it's a 9 plus 0. In the main part of the cilia, it's a 9 plus 2. Okay. Okay. Want to show you one other picture here. When the cilia actually move, there's two like phases. So when there's actually the power stroke, the cilia is kind of rigid. So now it would move, and it would move any debris or water up over here or fluids. But then when it returns back to its normal position, it becomes kind of soft, and it'll go back to its normal position. But then when it's gonna move substances, it kind of stiffens up and will form this power stroke. Okay. So we got the cilia. The next structure is the flagella. Now, in human anatomy, the only cell that has a flagella is in the male. That is the sperm cell. And its job is to propel the sperm up through the female reproductive system to get to the oocyte for conception. So here's a picture of cilia coming off the top of a cell, and then if you've never seen, here's a sperm cell. It has like a head, right? It has like this neck and middle piece right over here, and then this this tail right here, whip-like tail, that's the flagella. Okay, in females there are no cells that have flagella, only in males, and that's on the sperm cell. So this is, I'll let you kind of look at this on your own, but this is just an overview, kind of a review table here, showing you things that are in the cytoskeleton, the microfilaments, the intermediate filaments, the thick filaments. We're going to talk about these when we do muscle. I didn't really talk about them today, but these we're going to see in muscle cells. Microtubules, centrioles, cilia, and flagella. Gives you a little information about their location, and if you want to learn a little bit more about their functions. Okay, the next non-membranous organelle is probably one of the most important ones. We're going to look at this in subsequent lessons. We're going to look at it because these are involved with making proteins. Highlight right here, synthesize proteins, right? Synthesize means to produce, to make. Okay, when we look at a ribosome, we're going to see it has two parts, and we're going to get more into this later in the semester. This smaller part down here is known as the small subunit, and then look at this big thing over here. This whole big thing here is the large subunit. So here they're showing the two subunits together. This is what we have now as a functional ribosome. There are times when these are actually apart, and we'll talk about that later in the semester. Right, so there's two parts, the small and the large subunits, right, and they're made up of RNA, known as ribosomal RNA, right? I had mentioned this in our chemistry lessons. There was 3 types of RNA, and this is the ribosomal RNA that makes up the ribosomes. Now, we can find ribosomes within the cytoplasm. They can be free, just kind of randomly about in the cytoplasm, or they could be attached to something known as the endoplasmic reticulum, right? A lot of times we just abbreviate here the ER. So when they're attached to the endoplasmic reticulum, we call them fixed ribosomes. Okay, free ribosomes, fixed ribosomes. But regardless if they're free or fixed, attached to something, they still do the same thing. Their job is to make proteins, right? They're going to synthesize proteins. Okay, so again, here's our ribosome. Okay, the last structure is known as the proteosome, right? The proteosomes are a little bit smaller than ribosomes, and they contain these enzymes known as proteases. Anytime you see Proteo like this is proteins, and an ACE is an enzyme, and their job is to break down proteins that have like that are abnormal. They're not normally functioning. So we don't want to have proteins that aren't normal inside of our cells. So the proteosomes are going to kind of break down and recycle abnormal proteins, right? They kind of look like dumbbells to me. Like here you can see they look more like dumbbells, where ribosomes are a little bit more round in shape. Okay, so that concludes this lesson on the non-membranous organelles. Again, remember guys, these are organelles that don't have their own cell membrane around them. In the next lesson, we'll take a look at the membranous organelles. These are organelles that do have a membrane.